Good afternoon, everyone, and welcome to our second session of our five-part series, Urology Speaks. My name is Alan Parton, and I serve as the director of the Brady Urological Institute and urologist in chief for Johns Hopkins Medicine. In a few moments, I will introduce our speakers for today's webinar, Drs. Faisal and Fletcher, who will be leading our talk entitled Prostate Cancer in the Black Community, Tumor Biology and Health Disparities. I'm grateful for their willingness to speak on such a timely topic and feel touched to see it. So many of you have tuned in to listen and participate. We all know that prostate cancer is the second most common type of cancer to affect men, but we also know that it disproportionately affects Black men and by extension their families. It gives me a great deal of hope that our conversation today will lead to even greater actions tomorrow in order to better serve the Black men in our community. I'm often reminded that the foundation of our research here at the Brady is the relentless pursuit of thoughtful and translational investigations and the dedication to improving the future of medicine for all. Those of you who joined us for our inaugural webinar in September heard me share the story of the Brady's inception. Philanthropy is what enabled the establishment of our institute, thanks to Mr. Diamond Jim Brady over my right shoulder here, who was a successful railroad company investor and a patient of Dr. Hugh Hampton Young, one of the forefathers of modern urology and a trailblazing physician from the Johns Hopkins Hospital. In 1915, Mr. Brady became a partner in discovery by making a generous gift to build and launch the Brady Urological Institute. So on behalf of our speakers, faculty, residents, staff, and patients now and into the future, thank you for being with us as we are honored to have this opportunity to connect so that you can hear directly from our experts and ask them questions. Today's webinar is being recorded and will be made available if you would like to pass the recording along to any friends or family who might have an interest in watching it. After Farzana and Sean's presentations, we will take your questions. Now, please use the Q&A button at the bottom of your Zoom screen to ask a question. We will try our best to answer as many as we can. And now I proudly introduce you to our speakers. Dr. Farzana Faisal is the chief resident for the Brady Urological Residency Program. She started her medical career at the Johns Hopkins School of Medicine as a medical student almost 8 years ago. She's worked closely with her mentors, renowned Hopkins pathologist Dr. Tamara Lotan, and Brady faculty alumni Drs. Edward Schaefer and Ashley Ross to investigate the genetics of prostate cancer. For her work in cancer disparities, Dr. Faisal received the Physician Scientist Award at the 2019 Johns Hopkins University School of Medicine Young Investigators Day. This is a deeply meaningful recognition, but I know that it is only the start of all that she will contribute to our field. Our second speaker is Dr. Fletcher, a second-year Brady resident. He joined the Brady residency program after graduating from Harvard Medical School. Prior to his residency, Dr. Fletcher spent a year under the mentorship of Dr. Kwok Dien Trin, focusing on health disparities in African-American men with prostate cancer at the Brigham and Women's Hospital. His work resulted in Dr. Fletcher receiving the Harvard Medical School's Rose Siegel Prize, which recognizes excellence in research on the relationship between the medical profession and the community. So I am immensely proud to introduce you to these two very accomplished individuals. One of my greatest joys as the director of the Brady is to witness the development of our next generation of urologic leaders, and I can confidently say that we will be in good hands. Farzana and Sean, the screen is all yours.
Thank you, Dr. Parton, for the introduction, and thank you to all of our virtual attendees today. We're extremely grateful to all of you, especially our patients who are participating in this lecture series and encouraging our Brady mission of excellence in research, education, and patient care. I'm Farzana Faisal, a 6-year resident at the Brady.
And I'm Sean Fletcher, second-year resident at the Brady.
One of the strengths of the Brady Urology Residency Program is the access that residents have to groundbreaking urology research, as well as the endless research resources and support to conduct that research. In fact, we spend a full year of our residency engaged in scholarly research in any particular area of urology that interests us.
And as Dr. Parton mentioned, Farzana and I have become interested in understanding the disparities that exist in prostate cancer care. And we both spent time in medical school and residency investigating how prostate cancer disparities affect the Black men in our country. This is a critical topic in healthcare right now, and we're very fortunate to be able to present to you today. So why is this research important? It's been consistently shown that Black men are about 1.5 times as likely to be diagnosed with prostate cancer and over twice as likely to die from prostate cancer compared to white men. The tables below from a US National Cancer Registry demonstrate this fact. As you can see on the left, Black men have a higher rate of getting new cases of prostate cancer, and on the right it shows that they have a twice higher rate of dying from prostate cancer compared to white men. This has garnered considerable attention in the national media and is a pressing issue in healthcare. The reasons for this are multifactorial and involve differences in both tumor biology as well as disparities in access to care. Let's test our prostate cancer knowledge. So before we begin our presentation, we'd like to go over a few questions and involve the audience in answering a few things about prostate cancer. These are meant to jumpstart the discussion, and we'll address the answers in further detail during our talk. All right, so let's start with the first question. Number one, in what areas has prior research documented racial disparities in prostate cancer? Screening, diagnosis, treatment, or all of the above? We'll give a few seconds for everyone to answer the question. All right. I'll go through each one of them. Number two. Oh, sorry, the poll has appeared. Okay, I'll go through each question. All right, number two, one known difference in prostate cancer biology between Black and white men relates to tumor size, tumor location, the number of tumors, or all of the above. And lastly, Black men constitute what percentage of prostate cancer clinical trial participants through 2014? 3%, 5%, 10%, or 15%? So I think the poll ended a little bit early, but just to go quickly through the answers, I'll say them now and then we'll talk about them a little bit more throughout the talk. For number one, the answer is all of the above. So both screening, diagnosis, and also treatment have affected racial disparities in prostate cancer. For number two, the known difference in prostate cancer biology between Black and white men relate to tumor size, or sorry, tumor location. Tumor size, what the audience picked the most. I guess after all of the above is what they picked the most, but tumor location is actually the answer. And lastly, Black men constitute what percent of clinical trial participants through 2014? That answer is actually 3%. All right. So now that we have that knowledge under our belts, let's move on to a roadmap of today's presentation. Farzana will give an overview of the natural history of prostate cancer outcomes in Black men, and will also detail differences in tumor biology that may drive these outcomes. I will then discuss how social determinants of health and barriers in access to care may contribute to the higher mortality rates seen in Black men with this disease.
So to understand prostate cancer in Black men, I want to give you a closer look at the cancer outcomes in this group, particularly in the Black men who've been treated here at Hopkins. We're fortunate to have one of the largest prostate cancer patient databases in the country, and more importantly, one of the largest cohorts of Black patients. Going back to the early 1990s, we've had about 16,000 white men and about 1,600 Black men who've been treated surgically for prostate cancer at Hopkins. And we've consistently shown that at the time of cancer presentation, Black men are younger, they have higher PSA levels, higher Gleason scores and stages of their cancer, and overall, they just show up with much higher risk disease based on the National Comprehensive Cancer Network risk categories. And when these same men underwent surgery, Black men still had higher Gleason scores in their prostate specimens compared to white men. And Black men's Gleason scores were actually more likely to be higher than what was originally seen in their own biopsies, what we call upgrading. Again, Black men had higher stage disease than white men, so more likely for their cancers to have spread outside of the prostate. And then several years after surgery, Black men were more likely to have biochemical recurrence of their disease, even when taking into account socioeconomic factors like education and income. This suggests that tumor biology may likely be playing a role in these disparities. In fact, if you look closely at recurrence outcomes by race, Black men who have supposedly low-risk cancer, who are represented by the red dotted lines in these graphs, they have worse cancer recurrence outcomes compared to white men with similarly low-risk cancer, who are represented by the dotted green lines. And actually, Black men with low-risk cancer are more on par with white men who were deemed to have higher risk cancer, who are the solid blue lines on the graphs. So what this tells us is that race itself puts Black men in risk categories higher than what the conventional guidelines would suggest. And Black men really should know these risks and should be counseled that they may need more aggressive treatment plans compared to their white counterparts. And the same trend is seen for Black men with very low risk disease who are on our active surveillance program. While they're on surveillance, Black men are more likely to have disease progression compared to white men, meaning that on their subsequent prostate biopsies, their tumor grade or tumor volume increases and bumps them up to a higher risk category, which is represented by the red lines in these graphs. So overall, what this data suggests is that Black men may have more aggressive tumor biology. So to truly understand these differences, we need to look at the biology of prostate tumors in Black men. What are the ways we can study tumor biology? Well, at Hopkins, because we have one of the largest national databases in prostate cancer in Black men, we've been able to look at their tumor biology at many different levels. First, we can look at the prostate specimen as a whole after they've been removed from surgery and see if there are any differences in the tumor distribution or size between races. From the prostate specimens, we can also examine tumor cells to determine if the tumor cells exhibit a distinct molecular subtype in Black men. And then going even further, we can extract the DNA from tumor cells and perform genomic sequencing to see if there are any particular genes that are unique to Black men with prostate cancer. So with this research framework, we've been able to discover some key biologic differences in the prostate tumors of Black men. First, when we looked at the prostate specimen as a whole after it was removed during surgery, we found an interesting difference in the locations of tumors between white and Black men. Prostate cancer in Black men were much more likely to be in the anterior prostate or the front part of the prostate, about 50% in Black men versus 29% in white men. And this particular location of the prostate actually has drastic implications when it comes to diagnosing prostate cancer. The anterior prostate is actually very difficult to feel on a digital rectal exam, and it's also difficult to sample during a transrectal biopsy. As you can see in this diagram, the biopsy probe is right next to the back or the posterior of the prostate, so the biopsy needle can really only thoroughly sample tumors in the posterior prostate. The anterior prostate, which is up top here, can be missed. So if patients have tumors in the anterior prostate, those tumors can be easily missed, they can grow unrecognized, and subsequently they can be the reasons why Black men are presenting with more aggressive cancer. Fortunately, once we published these findings several years ago, we also saw concurrent advancements in MRI imaging of the prostate, as well as newer prostate biopsy techniques, which has allowed us to better target the anterior prostate in Black patients and subsequently detect these tumors as soon as possible. Secondly, we've been able to identify particular molecular subtypes of prostate cancer cells in Black patients. So molecular subtype refers to RNA or protein particles within cancer cells that may contribute to an aggressive cancer phenotype or behavior. Using our comprehensive database, as well as our collaborations with other institutions, we've been able to compare the frequency of different tumor molecular subtypes between Black and white men. And these graphs show that several molecular subtypes were found to be unique to Black men. Specifically, 6 molecular subtypes showed statistically significant differential expression by race. And these subtypes, just to understand, they're essentially molecules that are found within prostate cancer cells, and they're involved in many different cellular pathways like cellular growth, invasion, migration, and these pathways are hypothesized to contribute to an aggressive cancer behavior. So if Black men are more likely to harbor these particular subtypes, it would make sense that these subtypes could be contributing to the worst outcomes we're seeing in Black men. And remember when we said that Black men are more likely to have anterior prostate tumors? Well, we were also able to look at molecular subtype by tumor location in both Black and white men who've been treated at Hopkins. And we found that anterior tumors had less expression of androgen or testosterone receptor signaling, particularly in Black men. And interestingly, using a combination of different molecular subtypes, such as testosterone signaling, we found that anterior tumors had a higher Decipher score. So the Decipher score is just a calculated score that predicts the risk of future metastasis for an individual. So overall, this data and these figures suggest that molecular subtypes that are found in anterior tumors, which we know are common in Black men, are associated with worse prostate cancer outcomes. So taken together, all of this data really means that molecular subtypes in Black men are associated with a distinct genomic fingerprint. So what we need to do is to get even more granular and really look at the genomes of these cancer cells. It's undeniable that genome sequencing has been one of the most impactful scientific discoveries in the past few decades, starting with the Human Genome Project, which was completed in the early 2000s. In urology, a comparable revelation in our field was the genome sequencing of hundreds of patients with prostate cancer. And this was first published by the Cancer Genome Atlas Network, or TCGA, in 2015 in their journal called Cell. And this study identified the most common genes driving prostate cancer. This groundbreaking study sequenced 333 prostate cancers, but shockingly, of these 333, only 43 were identified as tumors from Black patients. And that, unfortunately, has been the trend since 2015 in the publication of genome sequencing studies in prostate cancer. Black men are grossly underrepresented. Despite the very well-known data that Black men have worse cancer outcomes, worse survival outcomes from prostate cancer, they continue to be grossly underrepresented in genome sequencing studies. Even after the TCGA publication in 2015, only a few other groups have really tried to include Black men in prostate cancer genome studies. When I started my research year as a fourth year, 2 years ago, there were only 4 other publications that had genome sequencing data on Black men, and the largest of these studies included only 90 Black men. So my goal during residency was to do a genome sequencing study using the largest cohort to date of Black men with prostate cancer, and to try to elucidate the key molecular drivers and genes that have been associated with poor prognosis in this population. Under the guidance of one of our pathologists, Dr. Tamara Lotan, our group has recently performed tumor genome sequencing in 205 Black men, and we characterize the most common genetic mutations seen in these men. And this table just depicts the most common genomic alterations that we found, and those occurred in the genes SPOP, FOXA1, and CDKN1B. And because we have long-term follow-up for our own patients, we were able to see if there were any associations between these genomic alterations and the patient's long-term cancer outcomes, such as metastasis. And there were actually 3 different genomic alterations that we found in our Black men that were associated with a risk of future metastasis. These were mutations in the gene TP53, which is a well-known gene that has been associated with development of cancer, many different types of cancer. Secondly, a greater overall burden of copy number alterations, which just refers to gene amplifications or deletions globally in the tumor DNA. And then third, copy number deletions in the gene CDKN1B. And this finding was actually a novel, never before published finding in Black men. So CDKN1B encodes for a protein called P27. This protein is involved in cell proliferation, and the loss of this protein actually has been associated with poor prognosis in several other cancer types, but this was the first time that this gene was shown to be a poor prognosticator for Black men with prostate cancer. Genome sequencing also is not only applicable to the DNA of tumors itself, but it can also be applied to germline or hereditary DNA in order to identify any heritable gene mutations that can run in a family and then can put an individual at risk for developing prostate cancer. For example, we all know that BRCA2 mutations are heritable mutations that have been confirmed to be associated with developing breast cancer, ovarian cancer, as well as a very lethal prostate cancer. So one of our goals here at Hopkins is to determine if there are any heritable genetic mutations that are prevalent in Black families with a history of prostate cancer, and thus could be contributing to the disproportionately high risk of prostate cancer incidence and mortality in Black men. With the generous contributions from the Ambrose Monell Foundation, as well as our federal grants, we've made tremendous progress in our research initiative entitled Hereditary Prostate Cancer, Discovering the Genes Responsible, which is led by Dr. Bill Isaacs and Dr. Patrick Walsh, who are, of course, giants in this field. To date, their project has sequenced the genomes of over 1,600 Black individuals, and it's identified nearly 50 candidate genes with mutations associated with prostate cancer risk in Black men. And I've listed just a few of those candidate genes that have been recently identified, including the 8p24 site variation, which was just recently published in European Urology as a genetic variant that's common in Black families with prostate cancer, as well as in those diagnosed with prostate cancer at younger ages. But this field of hereditary prostate cancer continues to be ripe for future discovery. Our next steps in this project to truly help the Black community include matching these hereditary mutations to mutations seen within the tumor itself, to characterize the specific function of these genes and how they may contribute to cancer. It's also important to quantify the exact risk each genetic variant contributes to familial prostate cancer. And ultimately, the goal really is to include these genes in the routine genetic testing of prostate cancer and guide the screening, diagnosis, and cure of prostate cancer in our Black families.
All right. So in addition to differences in tumor biology, there are also several aspects of social determinants of health that contribute to poor outcomes seen in Black men. In this section, I will review how barriers in access to care have helped to maintain the mortality gap between Black and white men with prostate cancer, drawing on several examples from previously published data. There's a vast literature that attempts to explain the reasons why Black men have poorer outcomes in prostate cancer care. For this presentation, we'll be focusing on a few key points. One, the suboptimal rates of screening and follow-up for prostate cancer in Black men. Two, race-based differences in treatment patterns for prostate cancer. And three, the underrepresentation of Black men in clinical trials for prostate cancer. The prostate cancer diagnosis and treatment pathway consists of a few main steps. It begins with screening, which involves a blood test that measures the PSA level or prostate-specific antigen, a marker that can potentially indicate there may be prostate cancer present. Diagnosis, which can involve additional blood tests and imaging, but ultimately involves a biopsy of the prostate gland. And lastly, treatment, which can involve surgery, radiation, hormonal therapy, chemotherapy, or a combination of those. At each step, there are examples of racial disparities regarding who is receiving these services and who is not. The PSA test is a blood test that gained popularity in the 1990s as a method for screening for prostate cancer. At its outset, white men, depicted in the graph on the right as a top line punctuated by the brown circles, were much more likely than Black men to receive PSA screening, and also more likely than Hispanic men to receive PSA screening. Black men here are shown in the third line from the top, marked by the triangles, and then Hispanic men at the bottom. Fortunately, these lines have been converging over time. However, it's believed that this large initial gap in screening rates between races has contributed to men of color, particularly Black men, being more likely to present with advanced disease and ultimately to die of prostate cancer. Moving from screening to diagnosis, in general, an elevated PSA test is often repeated to confirm that the level is still high. If it is, a man will ultimately undergo a prostate biopsy to determine whether prostate cancer is present. This study, which pulls from a very large clinical trial in prostate cancer, revealed that Black men under age 65 with an elevated PSA were significantly less likely than white men to undergo a repeat PSA test, and were also less likely to receive a diagnostic biopsy. This is another finding that may contribute to delayed diagnosis of prostate cancer in Black men, which can translate into a higher likelihood of presentation with advanced disease and a higher likelihood of subsequent death from prostate cancer. There are also racial disparities seen in prostate cancer treatment. This is a study led by my medical school mentor, Dr. Kwok Din Trin at Brigham and Women's Hospital, which draws from a large national cancer database. It aimed to compare treatment rates in Black men and white men with localized intermediate or high-risk prostate cancer, in other words, cancer warranting treatment. Controlling for sociodemographic factors such as education, income, and insurance, as well as clinical factors, tumor grade, and PSA level. The study revealed that Black men were significantly less likely than white men to receive appropriate treatment, that being either surgery or radiation. This is shown in the graph on the left. Black men, depicted in the left set of bar graphs, had consistently lower rates of treatment compared to white men from the years 2004 to 2013. Despite the fact that treatment rates overall increased, there was a consistent gap between races, as you can see in the graph. When we break the data down further, looking at outcomes of over 220,000 men treated across over 350 treatment facilities, a large proportion, close to 40% of facilities, had higher rates of treatment in white men. Close to 60% had roughly equal treatment between races, and just 1% had a higher rate of treatment in Black men. These data stand in stark contrast to what you may expect given the data Farzana presented regarding the higher likelihood of Black men harboring more aggressive prostate tumors. This study suggests that despite that knowledge, Black men are still being treated at lower rates than white men. Disparities in treatment are also seen based on the type of hospital a patient is treated at. In general, healthcare for racial and ethnic minorities is heavily concentrated in the US, with a small proportion of those hospitals caring for a large proportion of patients from these underrepresented backgrounds. Therefore, we conducted a study that focused on outcomes in minority-serving hospitals, defined as hospitals that care for a relatively large proportion of racial and ethnic minorities, specifically Black and Hispanic men. We used a large national cancer database to analyze treatment patterns among men with intermediate or high-risk prostate cancer, cancer warranting treatment, treated at either minority-serving or non-minority-serving hospitals. We found that patients treated at minority-serving hospitals compared to those treated at non-minority-serving hospitals were less likely to receive any treatment for prostate cancer. This is shown in the multivariable model on the left. The table also again demonstrates that Black men had lower odds of receiving treatment regardless of the type of hospital that they were treated at. Again, these data were controlled for several sociodemographic and clinical factors that are pertinent to prostate cancer care. We also compared the time from diagnosis to treatment between hospital types and found that this time frame was about 2 weeks longer in minority-serving hospitals, as shown on the graph on the right. This highlights another potential difference in treatment patterns that may be a marker of additional delays in care and suboptimal access to care for racial and ethnic minorities. I'd lastly like to mention the ongoing problem of underrepresentation of Black men in prostate cancer clinical trials. The graphs at the bottom right illustrate that Black and Hispanic men are largely underrepresented when looking at all types of clinical trials. The pie charts compare the national proportions of these demographic groups on the left and their corresponding proportions as a proportion of clinical trials on the right. In prostate cancer, the disparity is even more pronounced. Black men make up about, or Black patients in general make about roughly 12 to 13% of the population and make up only 5% of clinical trials. And in prostate cancer, Black men make up even a lower number, about 3% of all clinical trials conducted through 2014. The reasons for this are multifactorial. Some of you may be familiar with this country's unfortunate history of exploitation of racial and ethnic minorities in clinical research. This may have imparted a sense of reluctance of Black men to participate in prostate cancer clinical trials. Lack of access is also a contributor, with fewer resources being dedicated to outreach in communities of color. We now feel that it's imperative that we reach into these communities and engage them in understanding the stark disparities that exist in this disease and the need for increased participation in research to rectify them. Prostate cancer continues to exhibit some of the most significant racial disparities of any disease. As we've shown you, these disparities are present at every level, from tumor biology to cancer detection and treatment. The opportunities for research are ripe at every single level and are a key component of addressing a major cancer health burden in our country. So beyond the research, what are we doing here at Hopkins to address these disparities? One of the most important focuses here is translating our research findings into concrete actions that can improve the health of our local and national community. Here are just a few examples. We mentioned earlier the higher likelihood of Black men harboring tumors in the anterior or the front part of the prostate, which is more difficult to sample with traditional biopsy techniques. In our department here at Hopkins, we've refined and widely implemented the transperineal prostate biopsy technique, which allows for an easier and more accurate biopsy of the anterior zone of the prostate. This better equips us to find these tumors in Black men. Additionally, our work on genomics and molecular subtyping has allowed our pathologists to develop simple and cost-effective tests that we can perform on patients' prostate biopsy samples to look for molecular markers that may indicate risk for more aggressive disease. We've also taken strides in community outreach. While a lot of this is on pause due to the current pandemic, our department in general has an annual community booth at the Orioles game where we provide resources for prostate cancer education, and our faculty and residents also participate in a free prostate cancer screening fair annually at our Hopkins Bayview campus, and that's open to the entire Baltimore community. Community outreach continues to be an area in which we can do more for our local neighborhoods in Baltimore. We're fortunate to have colleagues at the Hopkins Bloomberg School of Public Health and the Sidney Kimmel Cancer Center who are epidemiologists, oncologists, and public health leaders who have expertise in cancer disparities. Our collaborations with them are another source of ongoing and potential work in this field. Lastly, we think we presented a lot of information, and I hope you all have a better understanding of racial disparities in prostate cancer care. To conclude, the opportunities to research and address these disparities in prostate cancer care are present here at Hopkins at every level. The ultimate goal is for us to increase the representation of Black patients in clinical trials and genomic studies of prostate cancer. And our research and your contributions will help to alleviate the pervasive racial gap of prostate cancer incidence and mortality outcomes. Thank you.
Thank you so much, Dr. Faisal and Dr. Fletcher. We're now ready to pose the audience questions to you and to Dr. Parton. So as a reminder to the audience, please use the Q&A button at the bottom of your Zoom screen to submit a question, and we will do our best to address as many as we can. There was one question that came in about the recording of this session. Again, it is being recorded, and we will be sure to make it available to participants after we have it. Please email bradydevelopment@jhmi.edu. Again, that's bradydevelopment@jhmi.edu. And now to get into some questions. How are the screening recommendations for Black men different from those of white men?
I can do this one. Yeah, so in general, there's actually been a lot of controversy for prostate cancer screening in general, but for the general population, it's currently basically recommended that for people of age 55 to 69 in the general population, that there should be a shared decision-making process with the patient and their physician about screening. There's no actual definitive age cutoff where there's a different recommendation for Black men. However, based on recent updates to these guidelines from several different national committees, they both indicate that Black men may be harboring more aggressive disease based on a lot of the data we've presented to you. And based on that, that they should talk to their physicians about screening earlier than that point, anywhere from age 40 to the mid 50s, essentially. So that's their current stance on it.
Thank you, Dr. Fletcher. Race is very much a social construct. How do you reconcile that with biologic and genomic studies in Black men?
I can take this one, Sean.
Right.
So I actually love that question. I think that's a very important question. We get it a lot, and you're absolutely right. Race is very much a social construct, and unfortunately, all of our biologic and genetic studies looking at the tumor biology of prostate cancer are really limited because race in these studies is identified by self-identification by the patient. And that's very much a personal and a social identification, but it doesn't take into account biologic aspects. So really the best way to truly incorporate race into genomic studies is to measure ancestry markers, ancestry genetic markers. Think about a hereditary test like 23andMe, where you get a spectrum of your ancestry genetic markers. That would be really the best way to incorporate race into our genomic studies. Unfortunately, this requires having access to and testing hereditary DNA, such as a saliva sample, not just tumor DNA, which unfortunately we did not have in many of our genomic studies. Though Dr. Isaacs and Dr. Walsh do have that data available in their hereditary prostate cancer biorepository, but that really is the next step for our groups and other groups around the country, is to look at race as ancestry informative genetic markers and see if there's any correlation between the spectrum of ancestry markers and outcomes of race, outcomes of prostate cancer. But the idea of race as a social construct is very important for more social studies when we're looking at social determinants of health, because race does impact, as Sean showed us in these many studies, patients' treatment plans, how quickly they get treated, where they get treated. So the idea of race as a social construct is important when we're looking at social determinants of health. Race as a biologic concept is important when we look at genetic studies. So that's a great question.
There's a question that says, can you have cancer while your PSA number is low?
Yeah, you can. It's possible. I think that in general, the PSA test is a good one, but it's not totally, 100% sensitive or specific, meaning that there's reasons other than cancer that can make it high. And even if you do have cancer, it's possible to still have a lower, on the lower end of the PSA range, a PSA value.
It's an interesting fact. When I was a medical student working under Dr. Walsh, we discovered that the higher the grade of the cancers got, the lower the PSA was seen in the men's serum, because for some reason, high-grade cancers have decided to quit making PSA because they have better things to do, such as dividing and spreading. So that causes some of this discrepancy that we see in high-grade cancers in men with low PSAs.
And on that note, it is, important to be able to monitor or trend your PSAs, even if they're low, with your physician and do shared decision making. We use things such as PSA density, PSA velocity. All of those things can really help us detect cancer, not just the absolute PSA number.
Doctors Faisal and Fletcher, you addressed the issue of racial disparities in the US. What about other Western democracies? Is there any research on that or any comparisons you can share?
That's a good question. To be honest, I think most of the data on Black men come out of the United States. I think some other, Western countries just don't have as robust data as we do. I have seen data come out of some African countries looking at genomics, which have identified similar genes that we have seen in our Black men. But I'm familiar mostly with just kind of the most robust data coming out of the US. I don't know if Sean or Dr. Parton, you're aware of anything else.
Yeah, I agree with that. I think the only other I've seen a decent amount from England, similar and really showing similar things to what we see in the U.S. But I think by and large, at least when it comes to looking more closely at social determinants of health, I've seen most of the things coming out of the U.S.
And I've had a chance to travel to Australia several times for scientific seminars, and I see the same results, exactly what we've seen here today amongst their studies.
The presentation noted that Black men who were tended to be younger. Is there an explanation for the age difference? And do we know?
Yeah, so sorry, I should clarify that. So when I said younger, they presented younger than their white counterparts, than white men. So unfortunately, Black men treated at Hopkins and nationally, they are more likely to be diagnosed with prostate cancer at younger ages. Hence the screening recommendation to start PSA screening actually younger than the age of 55, which is not really recommended for white men. So that just speaks to the biology of prostate cancer in Black men, is that they can be diagnosed with aggressive cancers at younger ages than other races.
This is a good follow-up question. What information can you share? What recommendations can you share about screening when there's a known family history of prostate cancer?
Sean?
Yeah. Family history is another known risk factor for prostate cancer. It's definitely known to very well track in family lineages. And that's something that in the national guidelines, I recommend as another reason, in addition to Black race, family history alone is a reason to talk to your physician about starting screening earlier, meaning earlier than age 55.
Do we know if any of these same disparities are present in Latino men?
So some of them are. So when it comes to treatment rates, so that's something that's seen actually similarly in Hispanic men, that they have lower rates of treatment compared to white men. However, when you look at the actual differences in mortality, the mortality rates of Hispanic men, that's actually closer to white men than to Black men. So I think that sort of speaks to the dual implication of both the biology of the tumor as well as the disparities in care. Different minority groups may experience similar disparities, but there are certain other things that makes it, the tumors may be more aggressive in Black men, and that may be why the mortality rate is higher. So sort of a combination factor.
A lot of our gap in knowledge there is in general, our inability to enroll Hispanic men in clinical trials. There's so much more we could learn, and that's also one of our interests here, is to improve enrollment.
Can you address what the financial burden of PSA testing may be for those who may have no insurance or suboptimal insurance? And might this be a factor in the lack of screening among Black and other minority communities?
I think I can handle that one. The PSA test in and of itself is not very expensive. I think if Dr. Lotan were on here, she'd tell us about $13 to actually run the test. But the problem is not the expense of the test, it's seeking medical care, showing up for a free screening, having a regular physician that you see. That's where our major gap is falling there. It's not so much how much the test cost.
And I think that also ties into our active surveillance program, because active surveillance really requires patients to see us every 6 months, get a biopsy every 12 to 18 months. And if patients fall off of that program, we're likely to see them 5-10 years later with very advanced disease. So especially with our Black patients on active surveillance, we have to keep a very close eye on their tumors, because they're more likely to become aggressive while they're on active surveillance. So keeping our patients in our programs and following them is very important.
So we have two related follow-up questions. The first is there an age or an average age where physicians should routinely refer their Black patients to urologists? And another question, given the potentially more aggressive nature of prostate cancer in Black men, is it even safe for them to be placed on active surveillance?
Yeah, so for the first question, I think it sort of harkens back to the first question we were asked, is about the screening rates. And I think that based on what the national guidelines show, really anywhere from age 40 to 55 is reasonable for a Black man to be referred to a urologist, or to even have just their primary care do the PSA screening. And then if there's an elevation, that's certainly a low threshold to then refer urologists for further workup. And then as far as active surveillance, and not just to go for the audience, is for men with lower risk prostate cancer, that may just warrant, instead of treating right away with surgery or radiation, tracking over time, as far as Anna mentioned, their PSA levels, getting a biopsy when it's indicated, when there's a rise in the PSA levels, et cetera. That is certainly can be safe for Black men. However, based on data from here at Hopkins, actually, and recent studies in the VA population, Black men have higher rates of disease progression on active surveillance and higher rates of getting treated for prostate cancer. However, when you look sort of at least about 7 to 8 years out, there's not a higher rate of metastasis and not a higher rate of death from prostate cancer. That's reassuring that it can be safe, but just with close monitoring, essentially closer monitoring.
Just want to also echo that you don't need to refer somebody to a urologist to get a PSA test done. It can be ordered by any physician at any visit. We did a large study I participated in, about 20,000 men, the Prostate Cancer Awareness Week data, where we found that a PSA of 1.5 in these younger 40-year-old, potentially African-American men was a useful number to refer to a urologist to think about. They're so much younger that a PSA of 4 is not a useful value for them. And to echo what Farzana said, seeing how that PSA changes with time is actually even more important.
Is there a difference in the type of care that minorities receive if they're in minority serving versus non-minority serving hospitals?
Yeah, so there's actually a lot of data on that actually extends outside of urology. So people started looking at this with just common medical conditions, heart failure, pneumonia, to see whether, and as I mentioned, the care for minorities is very concentrated. So there's a small percentage of hospitals that care for a very large proportion of minorities in the country. And when they look at the treatment patterns between those types of hospitals, they do see differences. So in urology, we see some, as I mentioned, in treatment rates for prostate cancer, bladder cancer, other conditions, and also different rates of death and readmission for things like heart failure, for pneumonia, et cetera. So there are definitely differences seen when you look at treatment patterns between those hospital types.
What are some successful strategies used to engage Black men when it comes to participating in clinical research?
There's a few. So I think really the biggest one is probably transparency and engaging the community as stakeholders and participants, not as just, that they're being experimented on, but just that they, presenting data to them, showing where the disparities lie, showing what the research question is, and really imparting the need for more participation in the research. Another big thing is making sure that recruitment sites and clinics where any study should be done are located in or very close to minority communities, as far as just reducing a barrier to access is a big thing. And then, accommodating things like work and childcare so that these patients are able to really participate in the continued follow-up that's required for, clinical trial participation. I think those are a few big things.
And there are, research grants funded by the NIH and by the government that are just solely focused now on Black patients and different diseases that really are only recruiting Black men or Black patients, which is something very new, and I think really going to help alleviate racial disparities in healthcare, because we're really concentrating on this population now at a national level.
What is present in the prostate that makes it more likely to grow cancer than other organs in a male?
That's a question. If we knew the answer to that, we'd be a lot further on. There is an interesting a concept that my mentor in the laboratory, Dr. Coffey, used to ask to every resident and medical student that came through here. As it turns out, the seminal vesicle, the organ that stores and produces quite a bit of the seminal fluid, is attached to the prostate, comes out when we remove the prostate, but almost never gets cancer. The same urine passes through it, the same blood flow passes through it, the same chemicals that we eat pass through it, but it almost never gets cancer. But the prostate, we know, is a high risk of getting cancer. And we have yet to figure out what it is about the prostate, but interesting concept that hopefully these two young people will figure out.
What are some additional examples of large-scale research efforts aimed at understanding the reasons for poor prostate cancer outcomes in Black men?
There are a few. There's one particular one that I know of is funded by the National Cancer Institute. It's called the RESPOND study, and actually Hopkins and Farzana's mentor, Dr. Lotan, is a participant. That basically stands for Research in Prostate Cancer in African American Men, defining the roles of genetics, tumor markers, and social stress. And basically their goal is to enroll a bunch of Black men, about 10,000 Black men, and really perform in-depth studies on genomics, on their actual tumor samples, and also looking at social determinants of health. And that's sort of a larger scale effort that's come, after a bunch of research has been done to build that foundation. So there's a few things that are in the pipeline. That's one of them.
And the same goes for, genomic studies, African American men. Hopkins is collaborating with so many other institutions in genomic studies and combining all of our groups and populations to get even larger sample sizes to see if genes that are prevalent in our patients are also prevalent in Black patients around the country.
I see a question in the Q&A there about the cell turnover rate in prostate. It's actually not higher. Farzana, you probably have looked at that. The cell turnover rate in prostate cells is probably slower than other organs.
Yeah, I would agree with that.
Has the incidence of prostate cancer in Black men been tied to lifestyle or other health conditions, or potentially even the work environment? Can you speak to this?
That's a great question, Sean. That's what you worked on.
Yeah, I think it's those are good questions, and it's a little bit harder to study. I think that there's a lot of ongoing research on particular things like diet, environmental toxins, things like that, that are maybe more prevalent in Black communities. I'm not sure that the data is fully borne out yet, but I think as far as, I think right now what we know of is driving the differences are largely due to treatment patterns. So, and treatment and diagnosis patterns, I should say. So I think delays in diagnosis, delays in treatment are the bigger things, but there's certainly a lot of investigation right now in diet, environment, which may also play a role, but I think that data just sort of yet to come.
I'll ask one last question and then we'll turn it over to Dr. Parton. Have we been able to test Black men for the new CDKN1B mutation that you spoke about in the presentation?
Yeah, so that's also a great question. As of right now, we're not testing Black men for the CDKN1B deletions. If you remember from my diagram, the prevalence of the CDKN1B deletion is still low in the Black population. In our cohort of Black men, it was only about 5 to 6%. So it's not common, but we know that it's dangerous. So testing for it at some point may be beneficial. Right now, genome sequencing of tumors is expensive. It may not be cost effective to test it in everybody, but the next step is to look for more cheap ways to do it so that we can incorporate it into screening for prostate cancer. One of the things that we did do on our study was to measure the P27 protein product of the CDKN1B gene. We can detect P27 in a much cheaper way. We can stain prostate tissue. That technique is called immunohistochemistry. So we were able to correlate the staining of P27 to patients who lost the CDKN1B gene. It wasn't a perfect correlation, so we still can't upscale that, but that really is kind of the next step if we want to start screening patients for this deletion, is to see if there's any other way we can do it in terms of cheaper staining techniques or using P27 as a proxy for that genetic mutation.
Well, great. In closing today, what a wonderful session. I just want a special thanks to Faisal and Sean for joining us this afternoon, presenting your research, and for answering so many of the excellent questions that came in from the group. I also want to thank everybody that tuned in today and participated in this webinar, and also thank you for your continued support of the Brady and Johns Hopkins Medicine. So also please consider to join us for other sessions in our series. Our next one is scheduled for Thursday, January 21st, where Dr. John Gerhardt, who's our Director of Pediatric Urology, is going to present on the clinical and life lessons he learned from over 1,000 bladder exstrophy surgeries that he's performed over his 40-year career here. So finally, thank you all again for spending time with us today. And I want to wish you a safe and healthy and happy holiday season. Thank you.